What if you could control brain cells with just a flash of light? It sounds like something straight out of science fiction, right? Well, today we're exploring the incredible science of photoneural interfaces. It's a field that's blending light, genetics, and neuroscience to literally change medicine as we know it. All right, let's get right into it. Just to show you how powerful this is, let's start with something that is frankly nothing short of a miracle. Right now, as we speak, a revolutionary therapy is using these very techniques to give partial vision back to people who have lost it. This isn't some far-off dream. It's happening today. And look, this isn't just an inspiring story. Clinical studies have actually documented this, calling it, and this is a quote, the first reported case of partial functional recovery in a neurodegenerative disease after optogenetic therapy. A sentence like that, that's a massive leap forward for conditions we used to think were completely irreversible.
So this all leads to a pretty big question, doesn't it? I mean, our brains don't have photoreceptors. They don't naturally respond to light. So how on earth is it possible to use a simple beam of light to control a neuron with pinpoint accuracy? Well, that's the central mystery we're going to unravel right now.
The answer, the key to all of this, lies in a field with a really cool name, optogenetics. This is the core technology that's making everything possible. It's the science of light control. So what is it really? Well, you can break it down by its name. First, you have genetics. You use genetics to make very specific brain cells sensitive to light. And then you have optics, where you use light to actually control those cells. When you combine them, you get this unbelievable millisecond-level control over individual brain cells. So, where do you get a light switch for a neuron? Well, this is the really clever part. Scientists actually borrow genes from simple organisms like algae. Yeah, pond scum. These tiny microbes have evolved these proteins you see here, called opsins. They're basically natural light-activated switches. And it turns out, we can take those very same switches and install them into our own brain cells. And it gets even better, because different opsins work like different kinds of switches. Think of it like a dimmer switch for your neurons. Some, like channel rhodopsins, are the on switch. You shine blue light on them, a channel opens up, positive ions rush in, and bam, the neuron fires. But others, like halorhodopsins, are the off switch. You hit them with yellow light, and they pump negative ions in, which silences the neuron, stopping it from firing. On and off. Okay, so that's the big idea. But how in the world do you actually install these little microbial light switches into the right cells inside a human brain? This brings us to the first part of the whole process, the genetics. It's this really elegant three-step process. First, scientists pinpoint and isolate the exact opsin gene they need. Let's say it's the on switch. Step 2, they package that gene inside a harmless, modified virus. Think of it as a microscopic delivery van. This virus, an AAV, has one job: carry the cargo. Finally, that virus is injected right into the target brain area. It finds the neurons, delivers its genetic package, and essentially gives them the new instruction manual for building their own light switches. So let's just trace that whole workflow one more time. You've got the virus carrying the opsin gene, it delivers that genetic code to a neuron, and suddenly, that neuron is photosensitive. From that moment on, you can shine a specific color of light and either activate that neuron or inhibit it, completely on command. All right, so the switches are installed. We're halfway there, but now we've got a whole new challenge. How do you get the light to those switches, especially when they're deep inside the brain, where sunlight, definitely doesn't reach? This is the optics half of the puzzle. And there are basically two main solutions, each one designed for a specific job. For the vision restoration we talked about, the target cells are right on the surface of the retina. So engineers built these amazing optronic goggles. A camera on the goggles sees the world, and then it projects patterns of light right onto those modified retina cells. But for targets deep in the brain, the solution is more direct. You implant these tiny, flexible probes that are packed with micro-LEDs. And these aren't just any old probes. I mean, they're a marvel of engineering. They're made of flexible, biocompatible polymers, so they can actually move with the brain, which causes way less damage over time. They can pack a ton of tiny light sources into a really small space, and get this, they can even combine light delivery with electrodes, so you can stimulate with light and record the result at the exact same time. It totally closes the loop. So we started this whole journey with restoring vision, which is obviously incredible. But as you can probably imagine, being able to turn specific brain cells on and off has applications that go so much further than just the eye. This technology is really poised to revolutionize how we understand and treat a huge range of brain disorders. I mean, just look at this list.
Researchers are using this right now to study the brain circuits involved in Parkinson's. They're figuring out how to stop the runaway neuron firing that causes epileptic seizures, or how to silence the exact pathways that send chronic pain signals. They're even using it to map out the basis of depression and memory loss from diseases like Alzheimer's. By finally getting this level of precise control, we can start to untangle what's cause and what's effect.
And this brings us to one final, really profound thought. For decades, we've had tools to read information from the brain, like an EEG, but optogenetics, it gives us the power to precisely write information into the brain, with the speed and the specificity of thought itself. So the question we're left with is, if we can now have a two-way conversation with the brain in its own native language of electricity and light, what comes next?